Did COVID come from a seafood market in China? Or did it come from the Wuhan lab? Or did it come from the United States of America? The former CDC director, Dr. Robert Redfield, suggested that the birthplace of SARS-CoV-2 could be Chapel Hill, North Carolina.
How strongly do you feel that this bioengineered versus a natural spillover, as
No,
they
I
say?
don't have any doubt in my mind. They're 100% confident that it was bioengineered. This virus was engineered. I think it was intentionally engineered as a part of a biodefense program. There is a real possibility that the virus birthplace was Chapel Hill.
Ralph Baric is a virologist at the University of North Carolina, Chapel Hill. He is the world's leading expert in coronaviruses and has been conspicuously quiet about his possible role in creating SARS-CoV-2, which led to the pandemic.
Coronaviruses are pretty good. I mean, you're a virologist, you know all this stuff, but you can manipulate them in the lab pretty easily. It's just spike protein drives a lot of what happens with the coronavirus, zoonotic risk. So you can get the sequence, you can build the protein, and we work with Ralph Baric at UNC to do this. Insert it into a backbone of another virus, and do some work in the lab.
First, let me give you some background information as to what Baric has been doing. Over the past 2 decades, Baric has received over $284 million in taxpayer grant money to conduct research on coronaviruses, among other things. The stated goal is to predict and get ahead of natural zoonotic spillovers. The unstated reason is for bioweapons research, also known as DURC, or Dual Use Research of Concern. It's called dual use because it can be used for both defensive and offensive military purposes.
What was the U.S. role in that?
I think it was substantial, and this is why, when you look at the accountability for China, their accountability is not in the lab work and the creation of the virus. Their accountability is not following the international health regulations after they realized that they had a problem, and allowing people like me at CDC to come in and to help them within 48 hours like they were obligated to based on the treaty. But the U.S. role was substantial. One is they funded the research, both from NIH, the State Department, USAID, and the Defense Department. All 4 of those agencies helped fund this research. The scientific mastermind behind this research is a guy named Ralph Baric at the University of North Carolina. And he was very involved in this research. I think he probably helped create some of the original viral lines. There is a real possibility that the virus birthplace was Chapel Hill.
The US government funds this research in China because they want to spy on China's bioweapons program. China allows the US to peek at what they are doing, not because they need our money, but because they need something else, our technology. More specifically, they need the US scientists' technological know-how. One technology that Ralph Baric and his colleagues developed is called seamless assembly, or the no-seam technique. This technique is used to alter viruses without leaving a trace of manipulation.
Se guardassimo il genoma della vostra chimera, capiremmo che era stata fatta in laboratorio?
Anything that we build in the laboratory has what are called signature mutations. It's like a little, it's where you signed your name, almost. It says, you put in these mutations and it says, this was built from material in the Baric laboratory.
Ma se non vuoi lasciare questa firma, puoi costruire in laboratorio un virus indistinguibile da uno naturale, giusto?
It is correct. You can do it without leaving a signature, yes. We're using any of the 3 or 4 different approaches for coronaviruses that were developed by different researchers. You can leave no trace that it was made in the laboratory.
In this clip, the reporter asks Baric, so do you rule out SARS-CoV-2's being a chimera made in a laboratory? Listen to his response very carefully.
Quindi lei esclude che SARS-CoV-2 sia una chimera fatta in laboratorio.
Not with the viruses that have been sequenced and reported to date.
In laboratorio possono esserci dei virus di cui non sappiamo nulla?
That's certainly possible.
Baric says, not with the viruses that have been sequenced and reported to date. He is very careful with his wording here. There are viruses that have not been reported that he and others have been working on that could be the progenitor of SARS-CoV-2. RFK Jr. says that the only reason for this technology is to create a bioweapon.
They took a virus that could only infect bats, and they made it now deadly to humans and made it infect humans very, very easily. They taught them another technique called the no-seum technique, and it's a technique for making the evidence of human tampering invisible. The only reason to develop that technique is for a bioweapon. It's the inverse of what you would want to do if you wanted to benefit public health. Everybody knew that this lab was not up to par, and that if you started doing these experiments at that lab, it was virtually guaranteed that something would ultimately escape.
Another technology Baric sent to China is called humanized mice or transgenic mice. In order to do experiments on how certain viruses affect humans, they take mice, strip away their immune systems, and then inject them with human DNA from stem cells from aborted babies. The same symptoms that manifest in the humanized mice are the same symptoms that would manifest in an actual human. Scientists then do experiments on the mice, infecting them with all sorts of chimeric viruses, and then see how it affects them. A breeding pair of these humanized mice was sent from Ralph Baric to the Wuhan Institute of Virology. Here is a clip of Stephen Quay and Jay Bhattacharya discussing humanized mice.
Biologists can take human cells, put them into mice. And so like the mice act in some ways biologically like human lung cells or whatever, right? That's a transgenic mouse that's humanized. And so you're saying in the diffuse proposal, they had demonstrated that it's very easy for humanized mice to pass the virus from one humanized mice to another humanized mice.
Yeah. So you take a mouse embryo at 16 cell or 32 cell or that sort of, and you introduce the gene for the human ACE2, which is again this receptor, and then you grow it up and you get a mouse and you get what's called a transgenic mouse, and that was done by Ralph Baric in North Carolina. And sometime around 2017, 2018, a breeding pair of those mice took a plane flight from North Carolina to Wuhan Institute, and a breeding colony was set up of transgenic mice there. It looks like those mice have now ended up in commercial business companies, biotech companies in China, where they're selling our American technology to other scientists. So that's a whole separate conversation. But yes, so these mice allow you to do serial passage to perfect the virus for the human ACE2 in a very easy way. And you can do one serial a week, and by 8 to 10 weeks, 2 months, you've got yourself a virus that is completely able to jump human to human from the get-go.
On March 24th, 2018, Peter Daszak from EcoHealth Alliance submitted a grant proposal to DARPA. This agency is part of the Department of Defense, and it develops new technologies for the U.S. military. The proposal was called Project Diffuse, diffusing the threat of bat-borne coronaviruses. The now disgraced president of EcoHealth Alliance, Peter Daszak, was to be the principal investigator of Project Diffuse. EcoHealth Alliance is the NGO through which NIAID and other U.S. governmental agencies funneled money to the Wuhan Institute of Virology. The amount requested for the project was over $14 million. In the DARPA proposal, Ralph Baric of UNC was to lead targeted immune boosting work. building on his 2-decade track record of reverse engineering coronaviruses and other spike proteins. Dr. Shizheng Li of the Wuhan Institute of Virology was to conduct viral testing on all collected samples, binding assays, and some humanized mouse work. In that proposal, it states that they were going to insert the furin cleavage site into viruses and then test the chimeric viruses on the humanized mice. We will analyze all SARS-CoV-S gene sequences for appropriately conserved proteolytic cleavage sites in the S2 and for the presence of potential furin cleavage sites. SARS-CoV-S with mismatches in proteolytic cleavage sites can be activated by exogenous trypsin or cathepsin L. Where clear mismatches occur, we will introduce appropriate human-specific cleavage sites and evaluate growth potential in varus cells and human airway epithelial cells. The furin cleavage site is what makes SARS-CoV-2 so infectious to humans.
I was concerned because of the presence of the furin cleavage site that we've talked about. And I think it's important to understand what that cleavage site does. That cleavage site totally changes the orientation of the binding domain of COVID. So it now, which could not see the ACE2 receptor, which is the human receptor, it totally changes the orientation now, so it has high affinity for a human receptor. So that furin cleavage site bothered me. It didn't seem that it belonged there. And then when you look at the sequences that it used, and it's beyond the committee, but I know many of you have looked into it, the sequences that they used in those 12 nucleotides for arginine were the arginine sequences, nucleotide triplet, that coded for the human arginine. So why did this virus have the arginine sequences for human there, not bat? It was very disconcerting to me. It looked like this virus was engineered.
Essentially, Baric was going to manipulate the viruses, and she was going to run the experiments on the humanized mice. The proposal claims this isn't gain-of-function research because they are not using pathogens that are dangerous to humans. These use bat SARS-CoV backbones, not SARS-CoV, and are exempt from dual-use gain-of-function concerns to infect humanized mice and assess capacity to cause SARS-like disease. The proposal was submitted March 24, 2018, but it was ultimately rejected by DARPA because they assessed it to be gain-of-function research. The proposal is considered to potentially involve gain-of-function DIRC research because they propose to synthesize spike glycoproteins which bind to human cell receptors and insert them into SARS-CoV backbones to assess whether they cause SARS-like disease. However, the proposal does not mention or assess potential risks of gain-of-function research. Nor does the proposal mention or assess dual-use research of concern issues, and thus fails to present a DIRC risk mitigation plan. Even though the project was not funded, some of the work may have been done before the proposal was submitted. Scientists often do preliminary work before a grant proposal is submitted. On March 8, 2018, before the Project Diffuse grant proposal was submitted to DARPA, Baric was asked in an email from Tony Rock, who was collaborating with him on the Project Diffuse. Hi, Ralph. I have a couple of questions about the SARS-CoV spike glycoproteins you are developing with respect to the DARPA grant we are collaborating on. This suggests that Baric had already started working on the lab work before the Diffuse grant proposal was submitted.
The Ralph Baric situation seems incredibly fascinating to me, that he would say, well, look, okay, I don't actually know if some of all, I can't say whether or not Ralph did some of this work that you're talking about that would have involved the furin cleavage site. Were you surprised that he kind of tossed responsibility over to his colleague there? And what's your guess on whether or not we're going to be able to get him to go on record?
Yes and no, because something similar happened in a debate. It was actually a very fascinating debate that was convened by Science Magazine in September over the origins of the pandemic. And the DARPA proposal had just been released ahead of the debate, so it came up. And one of Daszak's collaborators, Lin-Fa Wang, also kind of passed it over to Baric. And what's notable about Ralph Baric is that he signed one of the open letters by prominent scientists saying that actually a lab leak needs to be on the table and we need a full investigation of the origins of the pandemic. So, even he's one of the world's leading coronavirus researchers, if not the leading researcher. He has actually been fairly upfront about the risks of doing this research. And so I hope that he responds to our questions. We would love to interview him as well.
Yeah, that's a huge implication if it did potentially come out of North Carolina rather than Wuhan. And I think we should be investigating both leads, right?
No, I don't believe it came out of the university. Well, I don't think that's on the table.
So you're not even going to investigate that. I mean, that seems strange to me that you've got a guy who's saying that his colleague maybe actually did do this sort of research in order to get the grant money, and you're not going to even follow that lead?
He was saying that Baric's lab, so Baric was the one who wrote that part of the proposal and who proposed doing that work. To me, it's unclear where the work happened. And actually, I would cut this
So if it's unclear, then investigate it.
Right, so the work might have been done in Wuhan, right? Because they were collaborating with Wuhan, and so they're passing viruses back and forth. They're like, how does that work? Was there some of it being done in North Carolina? Or not this type of work, but is there a research lab there that could be a place that some of that work would be done?
Ralph Baric does have a lab that is working on coronaviruses there. That's correct.
Right, gotcha.
Well, Mara, thank you so much.
I would. Yeah.
You can finish your thought if you have something.
No.
Okay. Thank you so much for joining us.
Got to get Ralph Baric on. Dr. Baric, if you're watching, open invitation.
Ralph Baric told Peter Daszak in an email on January 13th, 2020, "Hi, Peter. I have to participate on an NIH call tomorrow at 10. I believe it's a strategic meeting designed to help craft an NIH response plan to the Wuhan coronavirus. Hope things are going well. Looks like we found our highly variable SARS-like coronavirus. Ralph." Fauci, in a deposition on November 23rd, 2022, perjured himself when he said he had never met Ralph Baric, except he may have met him at a conference where there are thousands of scientists.
Who are the last two authors listed on the top there?
Well, one is Ralph Baric, who I believe is a scientist at North Carolina who is a long-term grantee of NIAID.
Do you know Ralph Baric?
Not really. I know who he is. I doubt if I've ever met him. I may have met him at one of the meetings where there are thousands of scientists saying hi to each other, but I know who he is. He's a scientist at University of North Carolina.
And he's funded by NIAID?
He is.
But you don't remember ever meeting him in person?
Again, I don't recall. I could have met him. Again, I run into several thousands of scientists that we refer to, but I don't recall certainly having a relationship with him.
Baric was already on calls with the NIH as early as January 14th, 2019, and was emailing them earlier than that. On February 1st, 2020, Ralph Baric was on an infamous call with Fauci and other scientists to discuss the origin of COVID. On February 11th, 2020, Fauci personally met with Baric at NIH to discuss COVID origins and the P3CO, or gain-of-function experiments. So why did Fauci lie about meeting Baric? In an email exchange on October 17th, 2019, Ralph Baric, Tony Baric, his wife, Dashak, Shi, Lin-Fa Wang, and many others from EcoHealth Alliance were planning a conference call with Fauci's NIAID on October 30th, 2019 at 8 p.m. Eastern Time, 8 a.m. China time. In an email on February 25th, 2020, Emily Erbelding emailed Hugh Auchincloss stating that a request went back to Ralph for more details on how he might manipulate the viral backbones. This shows that the people at Fauci's NIAID were suspicious of COVID-19 being a manipulated virus. Dashak emailed Baric on May 27th, 2021, the following. Since we last spoke, I've checked a bunch of rules governing culture and viruses in the US, China, and other countries. Hope you don't take this the wrong way. Sending you this so you're aware in case you get questions from reporters and other scientists or the government agencies, etc. Not to disagree with your opinion, which I respect. In China, the rules allow for organizations to conduct culture of animal viruses at BSL-2, including chimeras. We checked with Zheng Li, who let us know that she used BSL-2 with negative pressure and appropriate PPE. I also know that they are stricter now on SARS-CoV. It's BSL-3, I believe. Ever since you showed it was able to infect human airway epithelial cells, so that's evidence they do take things more seriously, it would seem on the surface. I also checked the rules on a bunch of viruses for the US and was surprised to find lethal human pathogens cultured at BSL-2. For example, rabies and some vector-borne viruses, as well as many wildlife viruses. I also spoke with Chris Border, who let me know that bat paramyxovirus, cedar virus, close to Nipah hendra, is cultured at BSL-2, including the recombinants he has made with Nipah and hendra elements. Reference here. I've attached a list of some of the findings with references. Hope it's useful in case there are questions about this. I'm sure there are reasons for all of the above classifications and justifications that can be debated, but I just wanted you to know that I did the due diligence of this and checked that they were following the rules and that similar rules exist here. I'm sure it would be criticized. Maybe there will be tightening of biosafety levels given the hype around the lab leak hypothesis at the moment. However, I am still very confident that nothing untoward happened there, and have good reason for that based on the protocols they used, and the results they were sharing as we wrote a paper for Nature Communications in the lead up to the outbreak. Cheers, Peter. Baric responded, Sorry, Peter, you're being told a bunch of BS. BSL-2 with negative pressure? Give me a break. Their last paper mentioned BSL-2 with appropriate PPE. This last part was the first and only time this was ever mentioned. Never in earlier papers, and in the latest, never defined either. I have no doubt that they followed state-determined rules and did the work under BSL too. Yes, China has the right to set their own policy. You believe this was appropriate containment if you want, but don't expect me to believe it. Moreover, don't insult my intelligence by trying to feed me this load of BS. Ralph. This shows that Baric was upset that the Wuhan Institute of Virology in China was doing the experiments at inappropriate and dangerous BSL levels. So how do we know what work on this grant proposal was being done before the proposal was submitted? Well, we just need to politely ask the scientists at the Wuhan Institute of Virology and Ralph Baric to open up their books so we can see what they were doing. I'm sure they would be more than willing to do that in order to exonerate themselves and clear their names of any wrongdoing. Wrong. False. The WIV took its database of viruses offline in September of 2019 and has not been transparent. So we should expect better of scientists in America because they are so much more transparent than China, right? Wrong. False. Ralph Baric and UNC have been resisting FOIA requests for the past 2 years. Currently, Gary Ruskin at US Right to Know is suing UNC for them to release tens of thousands of documents that could help elucidate the origin of COVID. UNC has only handed over a handful of heavily redacted documents. Even Ralph Baric's wife, Tony Baric, tried to evade FOIAs by communicating via Gmail as opposed to her official email. This is an outrage. These are taxpayer-funded institutions and grants. We have the right to see what is being funded with our taxpayer dollars. Pressure needs to be put on UNC to release these documents, both social and financial pressure. Taxpayer funding of that institution needs to be halted until they hand over these documents. Donors need to stop giving to UNC until the documents are turned over. The Biden administration has not released all the intelligence on the origin of COVID, even though a law was passed unanimously in the House and Senate and signed into law by President Biden himself. In conclusion, there is culpability on both sides, China and the United States. From my vantage point, Ralph Baric could be responsible for creating the viral lines of SARS-CoV-2, and Shi Zhengli and her colleagues at the Wuhan Institute of Virology are responsible for carrying out these experiments at dangerously low biosafety levels, and it escaped out of the Wuhan lab either accidentally or intentionally, sometime in August or September of 2019. Thank you so much for watching, and please share this with as many people as you can.